Hello, this is a presentation on Chapter 11, Zebrafish.
So why study zebrafish? Zebrafish are cheaper than mice. They were only very recently added to the IACUC. They have transparent embryos. They breed all year and they produce many offspring. They have rapid development with 24 hours from blastodisc to embryo. And for mutagenesis studies, their embryos are permeable to chemicals, allowing for easy manipulation. This slide shows an example of how easy it can be to manipulate the zebrafish organism. This shows an in situ hybridization showing the expression of certain genes. As you can see here, the genes have an inflorescent tag attached to them which allows easily to view the genes as they are turned on. After the egg has been fertilized, it undergoes cleavage to go from one cell to two cells, and so on to four. This process is known as, in the zebrafish embryo, myrioblastic. So as these cells are splitting, they form what's called the blastodisc, this area right here, and this sets up the animal pole and the vegetal pole. So again, the term myrioblastic simply means that only a portion of the egg divides. Again, this portion up here. After a while, this begins to result in what's called a telo, meaning far, lecithal, meaning yolk, arrangement. So the cleavage is happening far from the yolk. So after 10 rounds of division, something special happens called the MBT. So the MBT stands for mid-blastula transition. Again, this occurs around division 10, and the primary result of this is these three layers: the external yolk syncytial layer, the enveloping layer, and the yolk syncytial layer. Some important changes are happening in the cells at this time. The zygotic genes begin to take over, so the maternal factors are no longer playing as big of a role. However, there are still channels between the cells allowing for movement of morphogens and maternal factors. After the MBT, the zebrafish begins the process of gastrulation. It is not birth, marriage, or death, but gastrulation, which is truly the most important time of your life, according to Lewis Wolpert. During gastrulation, the main products will be three distinct cell germ layers: the endoderm, the ectoderm, and the mesoderm. So gastrulation is a pretty active process. During this process, the embryonic shield serves as the organizer. So this shield, the organizer, is emitting signals that, over the course of time, transform the embryo into something more recognizable. After the formation of the three basic germ layers from gastrulation, dorsal ventral axis formation ensues. The site of the developing embryo with the embryonic shield becomes dorsal. So if on this, it'd be dorsal, and then in the other direction, ventral. So in this experiment, the experimenters took the embryonic shield from one embryo and implanted it into the next, allowing the formation of these two heads.